Hello, everyone. Can you hear me well? I hope. So my name is Camilo. I think half of you knows me already. I was a Putnam Fellow here. So today we are going to hear my great PhD student, Deniz. He is from Connecticut. He did his undergrad in Purdue. He studied political science and horticulture. And now he joined my lab and he is studying, also during Purdue, he worked in a project related with NASA and growing plants in space. So he was a great fit for my new lab. And now he's working on roots, spinach, root exudates. That was also inspiration from work that I saw here, the Arboretum. So with that, welcome, Deniz.
Make sure this one works too. Good, okay. Well, like Camilo said, I'm Deniz Camli-Saunders. I'm a third-year PhD student at URI and in Camilo's lab, and yeah, I'll be sharing today. So good afternoon. Thank you so much to the ARB for inviting me to present to you all today. I'm going to talk about something that is often overlooked in controlled environment agriculture, and that is roots. My presentation is entitled A Comparative Analysis of Root Morphology in Various Spinach Varieties, Field versus Hydroponic Growth Systems. As the title suggests, I'll be discussing how growth system, that is field soil versus hydroponics, reshapes root morphology and anatomy in spinach and what that means for optimizing cultivars and CEA systems. So the broader question that underlays this work is this. If we're working to redesign agriculture above ground through vertical farms and hydroponics, how are plants redesigning themselves below ground? Quite often we celebrate the technological sophistication of CEA with high-efficiency LEDs, automation, nutrient delivery precision, but at the core of all of that is still the plant. And so understanding how it responds structurally is essential. So controlled environment agriculture, or CEA, has emerged as a rapidly growing sector aimed at increasing production efficiency, reducing land use, and buffering environmental variability. By controlling light, temperature, humidity, nutrients, and oxygen, we create a highly stable production system. One of these crops that benefits from this is spinach. In many ways, CEA reduces environmental challenges, but notably, with the decrease in these environmental pressures, other variables begin to alter the plant. So how do these variables and traits become altered in these other systems? In all systems, roots regulate water and nutrient uptake, structural stability, and help to house microbiota. Traditional systems require roots to navigate things like soil structure, mechanical impedance, patchy nutrient distribution, and microbial complexity. Whereas in something like hydroponics, these exist in a relatively homogeneous nutrient solution with low physical resistance. And these are poorly characterized things in CEA spinach as of right now. Several studies have found that root systems do behave differently in hydroponic environments. Notably, the first among these were actually marijuana growers who ended up with giant root balls in their CEA systems. As stated earlier, there are several obstacles that a typical soil-based system must contend with. Compaction and a fight for nutrients is chief among them. Current research reveals that often plants grown in hydroponic environments are thinner, tend not to be lignified, and that when transferred from a soil-based environment, will actually opt to develop a new class of roots rather than continue with their soil-based structure. And we can see that here, and sometimes they grow into your sump tank themselves because of how prolific they are. There are limited amounts of data for inner anatomy, but there is some indication in lettuce and some cereal crops that conduction structures, that is our xylem, typically, are altered to expand to deal with the new development of structures such as these fine, highly branched root systems. You can see how dense and highly branched that gets. This is an unseparated scan in WinRhizo, which is one of our scanning softwares, of a root system at only 30 days old. We can see how many, probably hundreds of layers of roots there are, and probably 1,000 centimeters at least of root material just in that alone. So these profound differences are poorly understood in spinach, which is a main reason why we opted for this crop. Primarily, studies in this method have been focused on lettuce and herbs. Those that do develop into other crops have been limited to morphology, with root order and anatomy understudied. It's a time-consuming and difficult endeavor to study root anatomy, as there must be specialized tools, skills, and a weeks-long extraction process before several more weeks of slicing and staining and observation under a microscope. Those of you who have done sectioning will know how absolutely crazy this process is. And sometimes you end up with a completely unusable sample. So when we talk about root system architecture, we're referring to several things, including total root length, branching intensity, diameter distribution, and root order structure. These can roughly be organized into two broad functional groups. We have absorptive and transportative roots. First and second order roots are typically transport heavy, whereas third to fifth order roots are absorption and exploration heavy. We use the nomenclature laid out independently by two different researchers, Catherine Bellini and Jonathan Atkinson, who started looking into this about 20 years ago, and they ended up with a numbering system that goes like this. The most proximal root is your first order root, and as you go distal from there, it increases one by one all the way up to, in theory, seventh or eighth order roots, although you almost never see that in the wild or in our experiments because they're just so fine. They'll break off pretty much as soon as you handle them. There are other nomenclatures that actually go the opposite way. They go first order root as your most distal root, and coming back to the most proximal, they increase in order. So either one is fine. You just have to kind of specify what you're rolling with, and this is what we did. And this is a good way to kind of visualize that. You can see our first order roots coming straight off. Off of that is a second order root. Off of that is a third, a fourth, and so on. We did up to 5 in our experiment. And I also framed this study through the concept of root idiotypes. Now, a root idiotype is a theoretical model of an ideal root system that is optimized for a specific environment. For example, in a deep soil, a subsoil foraging idiotype might emphasize axial penetration, whereas in a nutrient-rich topsoil, something like dense lateral proliferation might be favored. The current model for an ideal idiotype in soil is the subsoil foraging idiotype, which is characterized by 3 things. First, a strong primary taproot, dense fine lateral roots, and is linked to efficiency and stress tolerance. And that was first described by Jonathan Lynch in 2013. And since then, they've seen this in corn, in beans, and several other crops, but have yet to kind of see that in a vegetative crop like this. So such a thing has not yet been categorized in hydroponics, but based on our prior studies, it does appear likely that perhaps fine root dominance or high surface area to volume ratio would be something that would be optimal. But do existing cultivars exhibit these kinds of traits? And how does growth system interact with things like genotype? Well, these are some things that we set out to explore. So our study had 3 primary objectives. First, to quantify morphological differences between soil and hydroponic systems. 2, to evaluate morphological and anatomical traits across orders, systems, and cultivars. And third, to determine impacts of system effects versus cultivar effects. And from this, we were able to develop some hypotheses. This is going to go like that. Okay. Some hypotheses. So first, that hydroponics would promote longer, finer roots, and we'd see a discarding of the lignification that we see in some of those soil roots. 2, that both morphology and anatomy would be highly plastic in response to growth systems, as we'd seen in some of the other crops. And third, that environmental changes would outweigh cultivar differences. So for our experiment, we grew 3 different cultivars, a fast, a medium, and a slow growth cultivar, aptly titled Income for our fast growth cultivar, Dark Side for our middle growth, and El Majestic, sometimes also known as Majestic for our slow growth cultivar. In both soil and hydroponic systems, these were NFT-based gutter hydroponic systems under controlled conditions in a walk-in greenhouse. We conducted periodic harvests on day 15, 30, and 45. All of those days involved morphological analysis, so we put our roots on a root scanner and analyzed them through WinRhizo. And then on day 45, 4 samples of each root order, so that's 20 per harvest, of each root order were collected for microscopic anatomical analysis. And our growth conditions were the following. We had a Crop King hydroponic system with a 12-hour photo period set to 250 micromoles per meter squared per second as our light intensity. We fertigated with a Jax Part A and B fertilizer, which is a 20-15-26 fertilizer, and had an EC of 1.6 and a pH of 5.5. For our soil, we did that in 1-gallon pots with a mycorrhizal premix, and that received 300 mLs of fertigation per day. We kept all that standard and same temperatures across for both of our treatments. And that allowed us to isolate things like system effects. So at our harvest, we collected fresh and dry biomass, which included fresh leaf and roots, which were then oven dried at 60 degrees for a period of 24 hours, and then reweighed, and that allowed us to contextualize our root traits. We also scanned over a dozen traits with our mycorrhizal scanners, and notably we measured root length, diameter, and surface area, and were able to quantify tips, forks, and crossings, which helped determine things like exploration of our root system. We also sectioned roots by order for our anatomical microscopy. Those roots were embedded in a JB4 resin, which is a plastic resin, which Ned knows plenty about. And they were sliced by our microtome at 1.5 microns before being stained with toluidine blue, which allowed us to isolate some of our carbohydrate-based structures. We then placed those under a Zeiss imager and analyzed their pictures in ImageJ, and I'll have some more pictures of that process later on. But here we can see in this above photo, this is the roots sliced down to their about half centimeter. They go into these blocks, they're filled with the resin, and then that goes under a vacuum or under nitrogen, and it hardens the resin. And then below, we can see this really nice 600x picture of this xylem with a very clear contrast of the blue versus the phloem next to it in the purple. So as for our analysis, we also had some 2- and 3-way ANOVAs. We had our factors of system, cultivar, order, and harvest, which made our R code very, very long because all of those needed to be individually labeled. And then for post-hoc testing, we had some Tukey's and some normality testing. So for an overview of our morphological results, we had 10 variables that we analyzed. As expected, we observed very strong harvest-based effects, which makes sense considering a 15-day-old plant looks radically different than a 45-day-old plant size-wise. But importantly, growth system had a stronger effect than cultivar in just about every variable that we measured. Here we can see our roots actually laid out nicely and separated by our wind rhizo scanner, and that's able to pick up our different diameter classes, which is really, really nice. But just to separate something like this out takes over an hour, and you have to pick at it with several sets of forceps and try not to scratch things up and break them. So, yeah. Then as far as our total root length went, I'm going to read out some about our analysis, and then I'll show you on a graph to make this a little bit easier. So importantly, our hydroponic plants exhibited significantly greater total root length, and that we saw this across all 3 harvests. So on our day 15 harvest, it's a little hard to see, and it'll be easier as we get bigger. So what you're looking at with this graph is we've got our 3 different colors correlate to cultivars. So our purple is our L. majestics, that's our slow growth. Our red is our dark side, that's our middle growth cultivar. And our income is our kind of tealish green looking cultivar. And then we further separate that out by growth system, right? So this striped that we see is our hydroponics, and our solid fill is soil-based. So what we're seeing in our first 2 harvests here is that by total root system length, hydroponics has significantly greater root length than its soil counterparts. And that continues and becomes even more obvious as we go along to our 3rd harvest, our 45-day harvest. So there were also significant system-based effects along with multiple 2- and 3-way interactions between systems, cultivars, and harvests. But the bottom line here is that by total root length, hydroponically grown plants are longer than their soil-grown counterparts. So continuing on to root diameter, Soil-grown roots are thicker on average than their hydroponic grown counterparts. So that is a reflection of something like mechanical resistance and the need for structural reinforcement and also for anchoring. Now here's what this really means if we dig into it. The thinner roots of hydroponics reflects the increase of higher order root development. That's third, fourth, and fifth order roots in comparison to soil, which is dominated by first, second, and third orders, which actually directly correlates to some of our earlier hypotheses about the types of roots we'd be seeing and the development of them because of differences in growth system. So what we're seeing, right, third, fourth, fifth of our root orders are thinner, and that is reflected here among all of our different graphs, all of our bars here. And El Majestic, our slowest growing, also had the largest diameter across all of our harvests. So not only does it grow slower, but it's also growing thicker roots overall. So for our fresh leaf weight, we had significant three-way interactions here. It's important to note, especially as we see from our second and third harvests, that we see significant results here, especially in our fast-growing cultivar Income, but also in our dark side, our middle growth cultivar as well. So we're seeing a strong main effect, but also interaction effects among this whole thing. But the bottom line from there is that morphological differences in our root systems are actually translating to shoot growth differences. And that, of course, is exactly what growers want, right? Who wants to grow less spinach, right, overall? And then if we look at dry weight, we're again seeing the allocation of resources to below ground root development here. In our second harvest, right, so our hydroponics is significantly greater with our dry root weight. And then that continues towards our third, with one exception, and that is notable, is that Income, our fast-growing cultivar, in the third harvest actually ended up with a smaller root weight than its soil counterpart. So it was literally producing more with less, which becomes important when we talk about things like cultivars for breeding going on in the future. We also have some additional traits here. From our tips, we have a cultivar-based effect. Our forks was system-based, but also harvest and system-based effects. And then we also have our crossings as well. So we can think about this as both a metric of root system development, right? The higher number of tips, forks, crossings you have, the more developed your system is overall. We can also think of it as a form of exploration, especially things like tips, right? If your cultivar wants to explore, it's going to be developing new areas and new tips for it to explore from. So when we visualize hydroponic and soil roots side by side, the differences become striking. We see that hydroponic roots are longer, finer, and more highly branched than their soil counterparts, which are thicker, shorter, and less proliferative. This level of architectural plasticity highlights how dynamic root systems actually are. Here on the left, we see this is the exact same cultivar. This is a dark side. This is our middle growth cultivar. Grown in hydroponics, they're both 15 days old. We see this very deep proliferative taproot, but also just this insane amount of branching. Already, we're seeing the development of third order roots even at day 15. Whereas you are definitely not getting that in this soil-based root, which seems to be lagging behind in its development. So as we move on from architecture to anatomy, I want to explain a couple things as to what we're looking at. So this is a cross-section, 1.5 micron cross-section of a first-order Income root. This is the first photo I ever took of a root. I somehow got very lucky that it looked nice. But for some structures, this is our xylem here. We have phloem next to it, which is more in purple than in blue. And then the entire thing is the root itself. This inner part, where all this vascular tissue is, we call that the stele. And that'll come in handy in the next few minutes. So our xylem are our hydraulic conduits, right? We have water running through them, and our sugars run through phloem. So when we were measuring anatomical traits per root order, we measured things like xylem number, our total xylem area, our xylem diameter, our root diameter, our stele diameter, and then also our vascular tissue ratio, which is just the steel diameter divided by the diameter of the entire root. But it's just a good way to visualize how much of the actual root is conductive tissue. So in our total xylem area, we saw significant three-way interactions. As we dropped down, we saw that our hydroponics did have slightly higher total xylem area as compared to some of our other ones, but that was not consistent across all orders and across all cultivars. It was just kind of a mishmash of data here, and we weren't seeing a trend emerge, right? In our lower orders, dark side was slightly larger than income, which was slightly larger than El Majestic, but that wasn't necessarily the same all throughout. In our second order here, like income was our largest and second order roots, but not necessarily the same across everything else. When we look at xylem diameter, we also again see a significant interaction, but there's no consistent system-wide trend here. We're seeing certain orders dominate from 1 to 4. Our order 5 differences are minimal, and stats are telling us yes, there's differences, but there's no way to quantify what exactly those differences are and say if we can attach this line trend to this, and we can predict this, but we can't. So then if we look at our steel diameter, this is a very fun graph that took a very long time to make here, and I'll explain it to you. So all of these circles represent a root. The inner circle represents the steel of each root. If the inner circle is blue, that's a hydroponically grown plant. If it's a gold, that's grown in soil. And then all the way on the left, right, when we see our purple, same color scheme as we saw before, that's our majestic. Our dark side is the red in the middle, and then income is on the right in green. But what we're seeing here is that there's some differences in our system-based things, but it's hard to look at it and go, yep, there's differences, because a lot of these are just looking the same. And when you're plotting them out through stats and through an ANOVA, it's spitting out that nothing's really different, right? Sometimes on some of these higher orders, for example, this income soil versus income hydroponic in our first order, we do see a pairwise difference there, right? But that's not consistent throughout the entire thing. So this consistency thing is a trend I hope you guys are picking up on. So then for some other interesting trends, as expected, our xylem number does decrease dramatically with increasing order, right? So a first order root has between 50 and 70 xylem typically. That's what we saw on average, and that drops down somewhere between 3 to 8 for our fifth orders. And that's expected, right? And same thing with your root diameter decreasing with order. And then we did have some interesting outliers. We had one vessel with 87 vessels in one of our roots. That was a dark side root. And then our smallest was all the way down to 3. And those are just like little itty-bitty babies. And then I also wanted to highlight the process of how we actually went about counting this. So we get a nice picture here from our imager, and then it's stained, it's great, we cut it, we can see it. Well, then we have to run that through a couple different softwares. So we have to then run it into ImageJ and give it a color code. So sometimes that involves actually coloring in our xylem so that we can get a nice difference and a contrast, and then tell the computer, this is the contrast, I want you to count that. Of course, we give it a scale and things like that, but it is a very interesting and in-depth process that takes hours and hours and hours to do 120 of these. So it just keeps going and going. And what does that leave you with? Well, it leaves you with this very large and very detailed amount. So this is showing all of our cross sections that we were able to capture for every single order that we had of our 3 different cultivars, and then in our different growth systems. So starting on top, that's our first order. We go down to second, third, fourth, and fifth. On our left side is our dark side again, hydroponics there, then soil, but dark side is next to it, and then income, which is our fast growing in the middle. But yeah, and you can see also the way the scale changes. We're looking at 950, almost a full millimeter here, right? And then as we drop down, we drop to 600 microns, and then 400, 250, and then right around 175 for our fifth order roots. So these are shrinking, and they're very, very, very small and very thin, right? You'll also notice that a lot of them look pretty similar, right? Roots are something that we expect to stay conserved anatomically, sometimes, apparently in this case, and especially down by fourth and fifth order roots, they really do look the same, right? So we know that that's not really changing. But what is changing is up in this first order, because for our dark side and our fast-growing income, they look pretty similar. And their steels take up pretty similar amounts of room in their cells. But something like L Majestic, which is our slow growth, actually looks radically different. It has a smaller steel, and it actually has this outer ring of xylem that goes outside of its steel, which was a very odd thing to look at and go, did this get folded on itself? Where did this random ring appear from? But it was there, and so it, of course, has to get counted. So here's where things get interesting. While morphology showed dramatic system-driven plasticity, anatomy was comparatively stable, and hydroponic roots showed slightly higher vascular tissue ratios. This may reflect subtle adjustments in transport efficiency under constant nutrient availability, but the magnitude of anatomical shifts was far, far smaller than morphological shifts and inconsistent. We also had some weak links to biomass, but it was really hard to get a grasp of what does this mean trend-wise. So, what does this mean? Well, one of the clearest findings in our study of spinach is that system effects exceeded cultivar effects. If we think back to our hypothesis, this answered our third one affirmatively. Environment shaped root traits more strongly than genotype, and we also had high root system plasticity in spinach. And that has implications for breeding, because if environmental context dominates, then selection strategies must be system-specific. We also have some morphological and anatomical coordination. Because despite dramatic architectural changes, vascular scaling relationships were preserved. And that indicates a coordinated developmental framework that maintains functional integrity even under environmental shifts. This suggests that while plants readily modify their external architecture, in our spinach varieties, internal vascular organization is more developmentally constrained. Continuing on with that, hypothesis one was that hydroponic plants would develop longer, finer root systems that did not have lignification-based thicknesses, and we clearly saw that. Hypothesis two included that anatomy would reflect these changes, but they didn't. We actually saw the opposite, where inner anatomy is conserved. And that differs from what other studies had found in cereal crops and lettuce. Now we also have one cultivar in particular, Income, which displayed traits that were strongly consistent with our subsoil foraging idiotype, which had been described previously only in soil, right? It exhibited fine root dominance and higher biomass. And that suggests that certain cultivars might be pre-adapted for CEA environments. Here we can see a scan of Income on day 30. We see this highly penetrating taproot that was there for probably since day 15, actually. They grow very quickly. But look at the thicknesses of these second order roots coming off of this. And then from there we can see third orders everywhere, right? So that is that finely branched root system that it was describing. So those findings suggest that root traits should be a prioritized feature in plant breeding. Two, that they're highly informative to substrate selection and root zone design and conditions. And third, that certain cultivars might be inherently better suited to CEA, which could improve yield and shorten growth cycles. Here is the exact same crop. One is in hydroponics grown and one is soil grown. Which one do we think is which? Soil on the right, actually the opposite. This is hydroponics here on the right, and this is our soil grown on the left, yeah. So you just, that huge fresh weight difference, right, both at 45 days. And I mean, this is a completely different plant, it looks like, than this, right? They're both sellable and they're both marketable, but if you're marketing this in bulk, your eyes light up like this as a grower. Okay. We, of course, do have some limitations like we do in all studies. We do have a small microscopy sample size, right, because of how time and labor intensive this is. Here we can see this was income soil getting prepped and ready to go. I just added paraformaldehyde to it and had to get vacuumed. And then the next day it was all the ethanol progressions all the way up till it sat at 70%, right? On the right, we have the hands of the wonderfully talented undergrad, who I'll talk about in a minute, trying to create a ribbon from our slicing of our plastic ribbons. So that just goes on and on and on. And our slicing took 4 months, right, just to get enough of those really, really solid samples. Then we also have some partial root loss during washing, which is inevitable. Here, this is a root that's been washed 3 times already, and there's still soil sticking to it. Those of you who do that kind of work will know it just never goes away. And that, of course, causes some breakage of some fine roots, which is why we have as many replicates as we can of that, because it kind of starts to balance out. As far as future directions go with this, we'd like to see expanded anatomical sampling. That just requires time. Some rhizosphere interaction studies in hydroponic systems, which would be very, very interesting. These have kind of yet to be characterized. And then also carbon allocation studies under elevated CO2 and various other environmental stressors, because all that CO2 increase is coming and it's not going to stop. With that comes drought, comes heat waves, several other things that we can't really deal with or have not learned to deal with yet. But there is tremendous upside to linking carbon allocation to growth systems. Here we see the start of kind of some of those things. This was a trial run of some exudate collection, and that same kind of style of collection can yield some of your microbes, and you can develop some populations off that. As far as stress response, this is a chiller we just got, which we're very excited about. So we're going to be looking at this same type of thing in different root zone temperatures, right? Because certain greenhouses can't necessarily control their root zone temperatures all that well, or they don't know if they should, right? Some other studies have come out about that, but they're all in lettuce, not yet spinach. So there's definitely some work to be done there. So to conclude, root morphology in spinach is highly plastic. Anatomical traits are comparatively stable, and growth system strongly influences root morphology. And certain cultivars exhibit idiolike-like traits that might be advantageous. So understanding and harnessing root plasticity is the next step in the future of this sector. It's important to know that roots are not passive. They are dynamic and responsive systems. If we want to optimize production in controlled environments, we must optimize the below-ground half of the plant. So I'd like to acknowledge a few people. So the Vijutta CEA lab, and in particular, our undergrad Ava Russell, pictured here with a tomato, for all of her help on this project. The Friedman lab, with thanks to James Fortin for his assistance in plastic sectioning and their willingness to teach me, because that is just very, very difficult. To the ARB itself for hosting, and to USDA NEFA for their support. Thank you again for the opportunity to share this work. And I look forward to your guys' questions. I do have a long references slide here. And then if you want to get in contact with either me or Camilo, you can do so, because our information's right here. All right. Yeah. So there's a few different angles. Lettuce as a market is saturated with growers already. There's a lot of people that don't necessarily want to get into it. Spinach is one of those crops that you find in pretty much every salad, which is nice. So there's a tremendous market there for it. But it also, because of its short life cycle and the root system that is similar to other things, kind of like kale almost, some of the findings that we have from spinach can be reflected towards other similarly styled root systems. And also there's so many varieties, right? So we can test for varieties that are been developed for certain environments, right? So our slow growth, our El Majestic, was actually developed for desert growth, which doesn't really make much sense. You need water in the desert, but that's very different than income, right? So income was actually soil-based, developed for soil, but it grows fast. Our one that was most optimized for hydroponics was our dark side, our middle growth cultivar. So and those have already been phased out of production. That's how fast that market kind of moves. So basically, it's our breadth of phenotypes that allows us to see all of these different results.
Yeah, I have a question. I came a little late, so you might have answered this already, but back in the 1970s, when I was working on nitrogen fixation,
Yeah.
we wanted to study the initial stages of it.
Mm-hmm.
We grew plants in hydroponics, and they did not have any root hairs.
Interesting.
Is that something you have observed when compared to soil? You haven't observed no root hairs, so we couldn't. So the microbe, the bacteria could not enter the root system. So we developed. I had to develop our own system. The aeroponics.
Mm-hmm. Yes.
Back to the 1970s.
Yes. Yes.
And all of a sudden they had root hairs.
Mm-hmm.
So I wonder if that's a trait that you haven't noticed that.
That wasn't one of the things that we focused on. They were present depending on certain circumstances, but definitely a lot less than in something that was soil-based.
You mentioned it, studying microbial interactions.
Yes.
Without root hairs, it's pretty hard for the microbes to relate to the plant.
It is, and based on some prior literature, we've seen that those root microbes are growing, so there must be some way for them to get there, but they just haven't been qualified or quantified as of yet.
For nitrogen fixation, they couldn't add any.
Yeah, yes, definitely not for like a mycorrhizae. Yeah, no.
Do you have any thoughts on why dark side seemed to do best of the two within the hypothesis? I think that's what you said, right?
Yeah, it was developed for it.
Oh, it was.
Yes, it was developed for it, but it didn't always do the best. It did the best in certain traits, but it wasn't. Sometimes it might have grown the largest, but it wasn't necessarily the most efficient, right? Thinking about like even the amount of root, the amount of carbon that's being allocated to roots, and then later to exudates. Depending on your root system, you might be less efficient, right?
So the biology is really interesting, and this question of what's really driving it in terms of roots. But as a breeder, you're ultimately just asking, how much shoot do I get?
Exactly.
So going forward, the breeding doesn't have to look at roots. Is that correct? You're just looking at yields, or and again, I'm separating this from basic biology.
Yeah.
As a commodity. How does that move forward? Do you just evaluate every cultivar out there with hydroponic conditions, and you dial in, what gives you the most dry weight in terms of shoot system, or is there something about the breeding where you would want to understand these roots as a
Yeah, I think the value of this is actually in lower tech CEA type systems, right? So you can take this knowledge and actually export it, because if you have something that has a more resilient root architecture, right, that's more resistant towards either droughts or from attack by insects or microbes, you can take cultivars that maybe they look like income or something that thrives in hydroponics, but maybe you take that and you can still grow it in, a very basic hoop house, right? But when a drought strikes, if it's got a more resilient root system, it might be able to weather that better. Or if it grows faster, you might be able to get 3/4 of a harvest out before some sort of catastrophe strikes. So I think there is a good amount of value in taking the knowledge that we have in our really advanced systems and using them in our less advanced systems that are more proliferative around the world.
So one quick follow-up.
Sure.
On the picture you showed of the two spinaches, hydroponic
Mm-hmm.
and soil, there was a visual quality. The soil-based plant looked darker green.
Yeah.
And so I'm just curious about the aesthetics in terms of your economic proposition. I would imagine most people would have said it was more desirable, I think, with spinach, to have the deep, deep greens that they associate with health.
Mm-hmm.
So that was just one picture,
Mm-hmm.
but have you assessed like chlorophyll content or
Um,
pigment contents, things that would happen?
That's forthcoming in our next few studies. Looking at like our temperature zone differences, we're going to look at some of the leaf-based measurements from there. But yeah, not yet. It's coming though.
Any other questions? Okay, let's thank him again and then go have some pizza.
Are we next spinach day?